All right, in this next video, we're going to talk a little bit about the processes by which water moves across the cell membrane, moves in and out of the cell, or the process of osmosis. So osmosis is just diffusion of water. But water is very important in life, so we speak about it separately, and we talk about how osmosis works in slightly different terms than we talk about diffusion. Diffusion of water from high concentration of water to low concentration of water, or oftentimes to describe it as the movement of water from an area of low solute concentration, the stuff dissolved in the water, to an area of high solute concentration. And again, the differences in osmosis we have to add across a semipermeable membrane. With diffusion, it doesn't require a membrane. With osmosis, it does. So the concentration of water is what determines the direction of osmosis by comparing concentration of water with the total solute concentration. So there's some terms we use to describe the solute concentration. If one solution has more solute, less water than another, we say that that solution is hypertonic compared to the other. Hypotonic, less solute, more water compared to another one. So water is going to move from hypotonic to hypertonic solutions. Isotonic means that the solute concentrations on both sides of the membrane are equal. So how do cells deal with water balance? Cell survival depends on balancing water uptake and water loss so the cell doesn't shrivel up or it doesn't burst. So if you put a cell in a hypotonic solution, an animal cell will swell up and burst. A plant cell, because of the cell wall, will create what we call turgor pressure. And that's important for the support of the plant. If plants don't get enough water, they wilt because there's not enough turgor pressure in their cells to support the cell structure. If you put a cell into an isotonic solution, the animal cell is happy. We use isotonic saline when we give IVs so we don't cause the red cells and other cells in the blood to burst if we gave them hypotonic, or shrivel up if we gave them hypertonic. In the case of a plant cell, that's actually going to cause the cell to lose some of its turgor pressure. If you put a cell in a hypertonic solution, the animal cell is going to crenate or shrivel up, which means it's not going to work as well as it should. And then a plant cell will go through plasmolysis, where the cell will kind of pull away from the cell wall as water leaves the cell. So again, here's a couple examples of where those things might happen to living things. So hypotonic, a cell in fresh water, higher concentration water on the cell, so the cell has to deal with water coming in all the time. So some protists, for instance, have contractile vacuoles to pump water out. Fish that live in fresh water, they urinate almost all the time to keep that water from causing problems with their cells. So that's an active transport that takes energy to pump the water out. So plant cells can deal with hypotonic solutions much better than animal cells in many cases. Here you can see the picture here. You can see the circle in the top picture there. That's the contractile vacuole filling up with water, and then you can see it in the bottom picture where it is contracted and pumped that extra water out. Put a cell in salt water, no matter if it's a plant or an animal cell, in most cases, unless it's specifically adapted to those environments, it's going to shrivel up and shrink. So they have to constantly take in water, and so fish that live in salt water, they don't pee very often at all. Plant cells will wilt. They can recover if it doesn't get too bad. Isotonic, again, no difference between the inside and outside as far as water concentration. The amount of water going in and out of the cell is going to be equal. The cell is healthy, it's balanced. Okay. That's why we talked about isotonic saline solutions. Plant cells are going to be less happy, but they can still be okay. So how does the water move through the cell membrane? Well, today we know, because of the work of two researchers, Peter Agri and Roderick McKinnon, that they have pores specifically for water, or water channels called aquaporins, that allow water to move rapidly in and out of the cell. Okay, so cell compared to the beaker in this case, the cell has a 0.3 molar solution, so and the environment is 0.1 molar, so the cell solution would be hypertonic. The beaker would be hypotonic, so water is going to flow from the environment into the cell.